Okay. B, everyone has said option B, protocells. Okay, great, protocells. Option B, very, very interactive audience. I'm loving this. Okay, option B, great. Should we wait for some more responses? Okay, so yes, great. So, yes, the answer is option B, protocells. Great. So now that we have learned about cells, okay, let's talk a bit about its discovery. Okay, how was cells discovered? Now, usually we talk about Anton von Leeuwenhoek and Robert Hooke, but today I'm going to talk about another person whom I think most of you have not heard about, or maybe you have heard about it. You can tell me in the chat box again. His name is Zacharias Janssen. Okay, he was a lens maker, and he was also the first person who is known to have created the compound microscope and also the first telescope. Okay, and once this, yes, Shai.
My name is Sheikh Adil Islam. I'm in 5th grade. I study in Alhorn High School. It is the person who invented the microscope and telescope. Like, which country is he from?
He's from Germany. Is that okay? Okay. So yes, so Zacharias Janssen, and what he did was, he created, so his compound microscope actually became the talk of the science town, and every scientist wanted to try their hands on it. Okay. And but so when this news spread that people can create microscopes which can actually magnify very, very small objects, what they so a lot of scientists wanted to try their hands on it, but there was this one Dutch scientist whose name was Anton von Leeuwenhoek. And he, instead of buying this compound microscope, he made his own microscope with sunglass lenses. And if you look at his microscope, it looked somewhat like the paddle of a boat. And if you place those two side by side, I bet it would have created a very cool sunglass, cool enough to create a fashion statement back in the day. And Anton von Leeuwenhoek, he started looking at different things under the microscope, under his microscope, including the plaque on his teeth. And back then, I did a little bit of research. I understood that people never brushed their teeth back then, which means he looked at lots of wriggling bacteria under the microscope, right? And he called them animalcules. And while he was looking at these different things under the microscope, he wrote a letter to one of his friends, okay, who was there in London, whose name was Robert Hooke. Now, Robert Hooke was kind of a jack of all trades, sometimes doing physics, sometimes doing chemistry, and sometimes putting his feet into biology. Robert Hooke, inspired by Anton von Leeuwenhoek, he also created his own microscope that looked somewhat like this. And with this microscope, he actually tried observing the cork of tree. You know, the outer bark of tree, like the cork. He made thin slices of the cork and observed it under the microscope. And he saw, what did he see? He saw something like this. Can you see, look at the compartments here in the cork? This looked like the compartments in the monasteries where monks lived. And that is, yes?
Is that thing stomata?
No, no, dear, this is not the stomata. So this is Hooke's observation under the microscope. Okay, so when
the first cell which Robert Hooke found was the onion cell, right?
No, it was not the onion cell, dear. It was the cork cell. So you have seen the plants, right, where they have a very thick bark outside, and he took the bark and he made slices of it. Like even like if you have seen lots of drinks have cork caps. He took that, he made a slice of it and observed it under the microscope. When he observed it, he saw this.
Okay.
So,
So like xylem, like the xylem vessels, you think?
so see, yes, you're right. So basically, cork is very matured, the xylem vessel with the secondary deposition of lignin and suberin, right? So basically, what he observed was dead cells, and it
Mm-hmm.
looked somewhat like this. As you can see, the cells have a very thick border. These are the depositions of cellulose and lignin. Okay, and when he looked into these cells, it to him it looked like these are the compartments in monasteries where monks lived, and that is where the name cell came from. So he is the one who named the cells. Okay, so Anton von Leeuwenhoek was the one to see cells for the first time. So if the question is who saw the first living cell, it's Anton von Leeuwenhoek, because Robert Hooke saw a dead cell, but who named cells? It's Robert Hooke. Okay, great, fantastic. Okay, now we are going to move on to the next question, and it's time to put your answers in the chat box.
And ma'am, it says some in the Google, it says in Google and also in our textbooks, it says that Robert Hooke was he's the one who discovered the first cell.
Yes, a lot of places say that. So the thing is that he discovered cells because he was the first one to call them cells. Anton von Leeuwenhoek, although he observed these living things under the microscope, he really didn't know them as cells. He called them
Oh,
as animalcules, as I just mentioned, because he saw the
Jules.
plaque of his teeth and he saw bacteria there and he didn't know what they were. To him, it was like wriggling creatures. So he was not the one who called it cell. So which is why books often say that, uh, yeah, he coined the name cell, right, Humera? Yes, good. Okay. Are we having the answers in the chat box, everyone, for the question that I just mentioned? Did you guys see the question? This is the question. Okay. Are we not having any answers in the chat box for this one? Is my
Yes,
screen sorry? Yes, Adan?
it's B.
Yes, it's B. It's B. Great, great. Yeah. Oh, I had already shown the answer. I'm so sorry. I'm sorry. I'm so sorry. I'm so sorry. Okay, we have another question. Uh, just a second. Okay, next question. Answers in the chat box, please. Okay. Manish, can you let me know if the chat box is filled? Has everyone answered? I would need some assistance here and there because I'm unable to see everything on screen. So I'm sorry, guys, if there is some confusion there. Okay. So observe dead cork cells. Fantastic. I think everyone has answered correctly, right? Great. Okay. Now, after the cells were discovered, there was some confusion about whether all organisms were composed of cells or where do cells come from. So that is when in Germany, there were two scientists, namely Schleiden and Schwann, who started their research. Schleiden was a botanist, whereas Schwann was a zoologist. So Schleiden was looking at different plants under the microscope, whereas Schwann was looking at different animal organs under the microscope. And both of them found that plants are made up of cells, and Schwann found that animals are made up of cells. So that is where the first two postulates of cell theory was formed, which are all plants are made up of cells and all animals are made up of cells. Now, the third postulate came much later when there was a fight between Schleiden and Schwann. So Schwann believed that cells came from pre-existing cells, but he was unable to convince Schleiden. That's when another scientist named Robert Virchow came into the picture. And he said that it has been scientifically proven that cells come from pre-existing cells because someone has seen it under the microscope, like someone has seen cells dividing under the microscope. And later on, we understood that Virchow was not the one who came up with the theory originally. He actually kind of borrowed it without permission from another scientist, Robert Rebec, if I'm not wrong of the name. Yes, so his research was a borrowed research. Okay, I think that is something new that we learned today. Okay, now time for the next question.
I
And
believe the
Manisha,
answer is Schwann.
who proposed? Yes, the answer is Schwann. Right, exactly. Okay, fantastic. Everyone is answering all the questions correctly today. Great, great. I'm so happy and so
Ma'am,
proud.
I didn't understand this question. Wait, what did Schwann propose that animals are made up of cells? Could you explain that to me, please?
Okay, so basically, Schwann was a zoologist and he was looking at different animal organs under the, as you know, zoologists work with animals. They cut up animals, take out their organs. And he was looking at a lot of the sections of these organs under the microscope. And he found out that all these organs were made up of similar compartments, which are cells, animal cells. So he is the one who proposed that all animals are made up of cells. Whereas Schleiden proposed the same about plants. Is that clear now, Shreyan?
Yes, ma'am.
Thank you. Thank you for your questions, Shreyan. Love answering those.
Thanks, ma'am.
Yeah.
No problem.
Okay. Now, based on the number of cells, okay, organisms can be of two types. Now, this is something that we all kind of know. There can be unicellular organisms and there can be multicellular organisms. Unicellular, meaning that are made up of one cell, multicellular, made up of many cells. But my question to you is, and for this, I would want you to think a bit and answer. My question for you is, why can't we multicellular organisms be made up of one single large cell? Why do we need to be made up of so many cells? Okay, while you think on that, I'm going to answer the question with a demonstration that too. But while you think on that, let me take you and let me help you learn a bit about the different varieties of cells. So as you can see, there is a whole new world of cells. As I said, there's a universe out there. It's a microscopic universe where cells come in different sizes and different shapes. Okay. And the size of cell and its shape, both of it is dependent on its function. Okay. We are going to come and explain how it is dependent on its function. But before that, do we have the answer to this question? Why can't we have one giant cell instead of trillions of cells? So, you know, the human beings are made up of 30 trillion cells. So why can't we just have one large cell? Okay.
Ma'am,
Yes.
it's because cells protects us from diseases, and diseases can like destroy our cells. Like one big cell is not enough to fight small diseases.
Okay. We are going to come back to the answer. What is everyone saying? Let me look at the chat a bit. Because our surface area to volume ratio would be too small. Yes, exactly. This is the correct answer, Sharanya. I'm going to explain this. This is fantastic. Sharanya, would you like to tell us a bit about this? Do you want to explain this concept to us? I can help you with some props here. Do you want to explain this concept to us, Sharanya?
Yeah, sure.
Yes, please go ahead. Tell me a bit about yourself first. Where are you from?
I'm from India. I'm living in the UAE currently.
Okay, fantastic. So yeah, Sharanya, go ahead. Let us all hear your explanation. I think you gave the absolutely correct answer.
So for unicellular organisms, they're quite small, so they have a large surface area relative to their volume, so like compared to their volume. So they can exchange like nutrients and oxygen just through their surface, through diffusion. And
Right.
that's enough. But for us, our needs are greater, so we need more of those substances. So we can't really do that efficiently through diffusion.
Beautifully explained, Sharanya, beautifully explained. I think I'm just going to add on to this explanation a bit with some live demonstrations that I have here. Can you see the boxes in my hands? Can you all see the boxes in my hands? I hope you can. Okay.
Yes.
So
Yes,
now, Sarath, can you help me a bit here with showing the boxes to everyone? Can we just place the camera in a way that we can see the boxes? And Mahira, could you please tell me if all the boxes are seen? Just give me a thumbs up, Mahira.
Yes, yes, we can.
Yes? Okay, fantastic. Okay. Let's look at this screen while I give you the explanation, okay? So this is one large box and it's a cube, okay? Now let's say this is one large cell, okay? And it has a side of 2 centimeters. So what would its volume be? 2 into 2 into 2, 8 centimeter cube, right? And what would its surface area be? One surface has a area of 4 centimeters square because 2 into 2. And for 6 surfaces, it is 6 into 4 centimeters square. It is 24 centimeters square. So what is the surface area to volume ratio here? It is 3:1. Everyone got it? Give me a thumbs up on the chat if you have gotten it. Give me a thumbs up on the chat. Manish, are we getting thumbs up on the chat here? Great, fantastic.
I got it, miss.
Yes. Now, let's take a look at the smaller cells. Okay. Now, if I break up this cube, into smaller cells, how many cells will I get? I will get 8 cells, right? I've broken up this cube into smaller cells, so I'll get 8 cells here. Okay, these are all DIY, so there is some measurement errors here and there. Do not mind that, please. So we are going to get 8 boxes. Yes.
Yes, ma'am. I've already typed in another example, another explanation for your question. Why not having one giant cell? Can you just check if that's also valid?
Yeah, sure. I will check it. I'll check it. Just give me a second. Let me just check it. Yes, absolutely. We are going to come to this. So you have given a final answer. This is going to be a conclusion. Absolutely. Shreyan, thank you so much. Okay. Now, these are the, yes, thank you. Thanks a lot, Shreyan. Now, these are the smaller cells. Okay. Now, each of these has a side of 1 centimeter. So the volume will be 1 into 1 into 1 centimeter cube. What would be the surface area? Okay, what would be the surface area? It would be 1 into 1 for one surface. So for 6 surfaces, it's going to be 6 centimeter squares. Okay. Now we have 8 cubes. So the total volume would be 8 into 1 centimeter cube. So 8 centimeter cube. But total surface area would be 8 into 6 centimeters square. That is 48 centimeters square, right? So what is the surface area to volume ratio here? It is 6:1. And a higher surface area to volume ratio means that the cell can exchange substances more easily and more readily. It can take in nutrients better. It can take in oxygen better, just how Shreyan mentioned. It will help in facilitate better exchange of oxygen, other gases, nutrients, etc. Okay, great, fantastic. Thank you so much, Sarath. Thanks a lot. Okay, moving on. Okay, moving on, we have a question here. Okay, again, Manish is going to tell me if everyone has answered, and then we are going to move on.
I believe the answer is mycoplasma.
Yes, yes, absolutely. Can anyone tell me what is the size of a mycoplasma average? Any idea? And one special characteristic about mycoplasma? Has everyone answered? Okay, let's see from everyone. Great, great. Okay, so mycoplasma, the special feature about mycoplasma, so the size of mycoplasma is something between 0.1 to 0.2 micrometers. That is 0.1 into 10 to the power minus 6 meters. That small it is, okay? And the special feature about mycoplasma is it has a fried egg-like appearance, okay? So these are also called as PPLO organisms, pleuropneumonia-like organisms, because they do not have a definite shape and their shape keeps on changing, okay? Great. Now cells of different shapes. Again, I would need some assistance of Sarat here. Cells of different shapes. If we talk about RBCs, right? If we talk about the red blood cells, it has a biconcave shape. Can you see? It has a biconcave shape. It's slightly hollow here. And the cells also lack nucleus. Can you tell me why? So there are two reasons here. I'm giving you a hint. There are two reasons here. Okay?
What is it used to for exchange of gases?
Sorry, could you please repeat your answer once?
What is it used to exchange the gases like in the bloodstream?
Yes, exchange the gases, but a little more specifically. Can we go a little more specific?
I'm like
So let's see.
exchange the oxygen or carbon dioxide.
Yes.
Um,
Okay.
And ma'am,
Basically, the major.
Ma'am?
Yes. Tell me.
I have another reason. Is it also because sort of exchange nutrients, air, water, minerals, stuff like that, vitamins?
Yes. So RBCs are responsible. The primary function of RBCs. Now RBCs are red in color because of a pigment called hemoglobin. Right? It's an iron-containing pigment which is red in color, which is why RBCs are red in color. Hemoglobin is responsible for carrying oxygen to different parts of our body, right? So now, if the RBC doesn't have a nucleus, then the oxygen molecules can fit so comfortably here, all cozy and comfy for the oxygen molecules, right? All cozy and comfy for the oxygen molecules. It can fit easily. Another reason is RBCs also have to reach depths of our body where we have very, very small capillaries, right? And which is why they need to squeeze in. Now, let's say this is it visible? Yes. Let's say this is a blood vessel. And let's say this is our RBC. Okay? With this kind of a biconcave shape, if I try to push it, see, the RBC can easily fold and squeeze into the capillaries, even though in size it is bigger. See, it has a bigger size than the blood vessel. Still, it can actually squeeze in and move in. But if it was a shape like this, could it move in? Can I squeeze it? No, right? So RBCs have a biconcave shape. And this biconcave shape helps it to squeeze into the capillaries, the narrow capillaries, supplying blood to different organs of our body and also fits in the oxygen. Great. Now I want to show you about something about the nerve cells as well. Manish, can you pass me on the nerve cell models, please? Okay, let's clean up the space a little. Now, nerve cells are supposed to be very, very long. Sarat, help me know a bit with this. Yeah. Thank you, Sarat. I'm going to introduce you guys to Sarat. Sarat is also from the Qtopia team, helping me here present this session to all of you, and Mahira. Mahira and Sarat has done phenomenal job helping me present this session. I'm going to do a bit on them at the end of the session. Okay, so wait for that as well. Okay. Now, are we getting a clear view of this? Sarat and Sarat, can you tell me, are we getting a clear view of this? Now, let's remove these a little here. Okay, we are done with these. We can get these removed from the table. Yeah. Now, we have a long nerve cell here. Okay. Let's remove this also, no? Yeah. We have a long nerve cell here. And we have two smaller nerve cells. Okay. So nerve cells are supposed to be very, very long. So can anyone tell me how the length helps it? What if I had two smaller nerve cells, two shorter, not smaller, shorter nerve cells instead of one long nerve cell? What would have been the problem? Can anyone tell me, or how does the long nerve cell help? Put
Ma'am,
down your, yes, yes.
ma'am, it helps the cell by so it will help it'll help it by communicating. If it would been short, it were not able to communicate.
Yes, exactly. So what happens is nerve cells communicate by electrical impulses. So electrical impulse passing, passing, passing. Then to jump onto the next nerve cell, for this electrical impulse to jump onto the next nerve cell, the electrical impulse has to be converted into a chemical signal here. Okay, so in between the two nerve cells, there's a space, there's a junction called synapse. And this is where electrical signal gets converted to chemical signal, which again, instead of converted to electrical signal to be passed on to the next cell. And this takes time. So if the nerve cell is long, one single long nerve cell, electrical impulse is whoosh. And then the next nerve cell. So we are reducing the time taken for information transfer. Okay. Now, at the end of this session, I am going to ask you why are muscle cells spindle shaped? So keep thinking on that. Okay. I've given you the answer to 2. So you are going to give me the answer to 1. Cool. Everyone.
Ma'am, can you repeat that question again?
The question is that why are muscle cells spindle shaped? Why are smooth muscle cells spindle shaped? Also, skeletal muscle cells are branched. Why? We can talk about muscle cells at the end of the session. Okay, great. Now moving on. Okay, we have done this. Now let's choose the correct answer here. And Manish, you will tell me whether we have gotten answers in the chat box.
I believe the answer will be function.
Yes, exactly. We just learned that the shape of the cell it depends on its function. Great. Now we are going to talk about the boundaries of the cell. The boundaries of the cell. Okay. Now, when we talk about the cell boundaries, the first thing that is, can you tell me what is common to all of these cells that you see here in the picture? In the chat box, please. What is common to all of these cells that you see in the picture? Put it out. What is common? Let's think on it. Let's put our thinking caps on and think on it a bit. Okay, someone says they have an atom. All of them have said, yes, cell membrane. Yes, fantastic. Cell membrane. Great, great. Nucleus. So nucleus is not correct because we also showed a prokaryotic cell, right? And the prokaryotic cells have a nucleoid and not a nucleus. They have a nucleoid, right? Okay. The cell membrane. So kudos and congrats to everyone who said cell membrane. Now, what are the different properties of a cell membrane? Now, the cell membrane, to understand its properties, we need to understand what is the structure of the cell membrane. Okay. And for that, okay, now imagine this. Also, Manish, with this one, can you get me the bread and the chocolate, please? Yeah. Okay. A fun demonstration coming your way for sure. But before that, the cell membrane is a lipid bilayer. So you have a layer of lipid on top and a layer of lipid on bottom. As you can see, it's a lipid bilayer. And the lipids have, if you have read this in chemistry or if you have read this before, you know that the lipids have a hydrophobic and a hydrophilic end. That means one part of it is polar, the other part is non-polar. To say it in a simpler language, one part of it loves water, the other part of it hates water. So hydrophilic end, the water loving end, and the water disliking end, the water hating end. Okay. So the circle part that you see is the water loving part, and the tail is the hydrocarbon tail. It is the part that dislikes water. Okay. Now it's a lipid bilayer. And in that bilayer, we have protein molecules. Okay. And this, because it is a lipid, right? If you look at even if your skin, okay, if you look at your skin is stretchy and it has it has, because of subcutaneous fat, right? So lipids are stretchy. So lipid basically makes the cell membrane a bit stretchy. Okay. Just give me 1 second, guys. We need the chocolate sauce from the fridge. Okay. Now,
Ma'am,
okay.
those purple, my bad for interrupting, but those purple
Yeah.
structures are
This is the
carrier
protein.
proteins, right? Active
Okay, so
transport.
there can be there are different kinds of proteins. Okay, there are different kinds of proteins. Some can be carrier proteins, some can also be aquaporins through which water can pass. Okay,
Aqua what?
aquaporins, AQUA, aqua for water, and porins, PORINS. Okay, and these
Okay.
proteins also enable a lot of molecules to pass through them, some important molecules, some important nutrients to pass through them. Okay, great.
Yes, ma'am.
Now, because this is a lipid bilayer and there are proteins into it, you can think of this more like these two pieces of bread. This is a layer of lipid and this is another layer of lipid. So you sandwich them and we get a bilayer here. Okay. And somewhere here we have proteins embedded through which molecules can pass into the cell. So for example, if this is my cell, okay, Sarah, can you help me a bit here? If this is my cell and if this is the cell membrane, then this is one lipid layer and this is the other lipid layer. Okay, so this is a lipid bilayer that you have. Now, because of this lipid bilayer, the cell membrane is also semi-permeable and selectively permeable, both together. Okay. Now, so some molecules which are small can pass through this lipid bilayer by the process of diffusion. I'll show you how. So let me pour this here. Now, if I am pouring things here, because this bread is porous, the sauce that I have poured on top will ultimately reach the insides of it, right? Which is why it will feel more soaked. Similarly, the cell membrane is porous, okay? Which is why gases like oxygen and carbon dioxide can easily pass through it. See, because of these pores, the sauce that I have poured can easily go inside, okay? And when I have it, it will feel soft and soaked up. And this is exactly what happens with gases in the cell membrane, okay? Great, fantastic. Yeah, thank you. Now, as I said, the cell membrane is semi-permeable. It allows only certain substances to pass through it. Now, what are these substances? Either small gases like oxygen and carbon dioxide or things, water can pass through aquaporins, and certain small, and the molecules that the cell needs basically to function properly. All of these can enter the cell through the cell membrane and the proteins embedded in them. Okay. Now we have a question. Can we answer this, please? Answers in the comments below. Not in the comments below, sorry. I forgot that this is not YouTube live. I'm so sorry. Answers in the chat box, please. But Mahara, it's also a YouTube live, right?
Yes, yeah.
Yes, great, great. Are you guys having fun? I want to know that. Are we having fun today learning about cells? Because,
Yes, ma'am.
yes.
Yes.
Okay, okay. That's really nice to hear.
Yes, ma'am.
Having fun. Okay, fantastic. Great. We have a lot of yes here. Great. And how many participants? Oh, we have 52 of you here with us. That's so exciting. I'm so happy to have all of you here. Once again, for the ones who joined a little late, a very, very warm welcome and a very good evening to each one of you. I hope you have a wonderful evening, a wonderful Saturday evening learning about cells. Okay, great. Are we done with all the answers? Okay. Can we see the screen? Can we have this removed, Sarat? And also this maybe. Yes. Yes, this also we can remove. Great. Have you gotten all the answers, Manish? To this question? No one has answered? Everyone has answered? Great, great, great. We have the correct answer. It's a bimolecular lipid layer surrounded by proteins. Okay. For some reason, my screen is stuck. Just give me a second. Okay. Now, functions of the cell membrane. This is where things are about to get interesting, right? The cell membrane helps, number one, it protects the cell. Number two, it helps in transport of substances in and out of the cell. It helps in the transport of substances in and out of the cell.
Ma'am, it's because it's semi-permeable.
Yes, exactly. It is because it's semi-permeable. I will need some water here. It's because it's semi-permeable. Okay, great. Now, about the different types of transport, right? There are two types of transport based on whether the transport requires energy or doesn't require energy. It's active transport and passive transport. So passive transport, name suggests, doesn't require energy. Active transport requires energy. Okay. Now, one of the most important examples of passive transport is diffusion, the way oxygen enters our cells. And I am here to show you how diffusion takes place, right? So, let me show you. Can you see my teacup here, everyone? Can you see my teacup? Okay. And I have some tea bags. So it's evening and it's Bitasta's tea time. So Bitasta is going to brew herself some tea. And let's see how diffusion plays its role here. So Bitasta is dipping it. Oh. Is the water not warm enough? Can we warm up the water, please? Just give us a second, guys. The water, the color is actually coming, but it's very less. I don't think you could see it. I want you to see it. So just give us a second. Let's warm it up. Okay, great. So diffusion is the way in which cells exchange oxygen and carbon dioxide, okay? And another form of, so this is exactly what is going to happen. I'm going to show you guys. And then I think I've already given you the answer to this. So let's skip answering this, I think. Or do you want to answer this? Do you want to answer this?
Yes, ma'am.
Yeah. What is the answer to this?
A.
Oxygen passes through the cell membrane with the help of the transport protein. Okay, oxygen passes into the cell with the help of diffusion, right? So it doesn't require transport protein. It can directly get in through the lipid bilayer. Okay, great. Fantastic. We have something in the chat. Yes, the answer is false. Exactly. Great, great. Okay. Now the next concept after okay, looks like my water is ready. So let's try this. Let me show you how diffusion should work. Okay. Now I'm dipping it. Can you see the color change slowly? Can you see it, everyone? Sarah,
Yes,
can you
ma'am.
focus?
Yes,
Yes.
I can see it.
This is happening because the flavonoids, the molecules in tea are diffusing into the surrounding water. Diffusion is a process by which molecules move from a region of higher concentration to a region of lower concentration, which is why it doesn't require energy to move. Okay, let us go back in the cells once. Yes, see, flavored molecules in the tea leaves move from the tea bag into the hot water. In the tea bag, they are concentrated. In the hot water, there are no flavor molecules from the tea. So it moves from a region of higher concentration to a region of lower concentration, without any involvement of energy. Okay, great. Now we are going to talk about osmosis. Now, osmosis is very, very similar to diffusion, except in osmosis, there is a movement of the molecules of a liquid across a semi-permeable membrane from a region of higher concentration to a region of lower concentration. Okay, through a semi-permeable membrane. See, molecules are moving from their region of higher concentration to a region of lower concentration. And in osmosis, when we talk about osmosis, it is very important for us to understand the different kinds of solutions that there exists. One kind of solution is like, say, plain water. Okay, say I have plain water here, and I have some raisins here. Can everyone see my camera?
Yes.
Yeah. And I put this raisins in this plain water. Now, raisins have highly concentrated sugar in them. Okay, so their solute concentration is very, very high. Whereas in the water, this is plain water, it is zero solute concentration. Okay, so what will happen? Water will move from, so water is going to move into the raisin and it is going to swell up, right? But what if we added the raisins to a solution that is highly concentrated in sugar? Okay, I've already added this from the very beginning. So what if we added raisins to a very concentrated sugar solution? Can everyone see my screen? Yes, concentrated sugar solution. What would happen? Water is going to move out from the raisin because the outside solution also has a very high concentration of sugar. So the raisins are going to look somewhat like this. See, it has become very, it has a lot of lines here because it has lost water. Can you see it? It looks somewhat like the pruned fingers. Can you see it? Can you see it here? Can you get the camera a little close, Sarat? Here? Can you see it has a pruned appearance? And whereas with plain water, right? The ones that I had put in plain water, if you look at this, these are all swollen and bubbly. Look at how swollen and fluffy this is. Okay, so a solution that has a lower concentration of solute, okay, is called as a hypotonic solution. Okay, and a solution with a high solute concentration is called as a hypertonic solution. Okay, and the solution which has the same tonicity, okay, or the same concentration of solute as the substance that you're putting in it is called as an isotonic solution. Okay, cool. Is everyone clear on that? Or do we have some confusions? Do you want to explain? Do you want me to explain again? Anything? Any questions?
Ma'am,
Yes.
ma'am, like how do does osmosis also happen in our body?
Yes.
Like many times when I put my finger in water for a very long time, it will get wrinkles.
Yes, that is because exactly because your cells are losing water. That's a really, really smart correlation you made there. Lots of love and kudos and congratulations for this. It's a very, very smart example. Yes, osmosis is taking place constantly within our bodies, even within our kidneys, osmosis is taking place. That's how we maintain the right concentration of solutes in our urine as well. Right? Great, great. Thank you so much for that. Okay.
You're welcome, ma'am.
Thank you.
You're welcome.
Okay. So what happens when we put raw mangoes are placed in brine? Brine is a very concentrated salt solution. So it's going to lose water, get wrinkled and shriveled. Okay. Okay. Now we have a question for us. Manish, do let me know when everyone has answered. Okay. Great. So we have gotten a lot of options. Yes. So both diffusion and osmosis requires a concentration gradient, okay? Because they do not require energy, and molecules move from a region of their higher concentration to a region of their lower concentration through a semi-permeable gradient, and which is why they require a concentration gradient. Great. Okay, now let's answer this question. It's a slightly more challenging, but I am sure you guys will ace it. All of you will ace it. You have not seen a single incorrect answer in the chat box till now. So let's see. Are we getting all the responses? If a cell
I
needs to...
believe it's active transport.
Okay. If a cell needs to move yellow particles across the cell membrane from A to B, which of the following will it use? Okay, everyone is saying it will use active transport. It will be passive transport, no, because the yellow molecules are higher outside the cell than inside the cell, right? Is that correct?
Oh, yes.
Yes, so yellow. So basically passive transport can only take place when there is a higher concentration of a certain molecule outside the cell and the same molecule is present in lower concentration or zero concentration within the cell. Only then will it move from a region of higher concentration to lower concentration. Okay, so it's going to be a passive transport. No issues. Great, great. Okay, now identify the solution which is more concentrated. By looking at the solutions, can you identify which one is more concentrated? And after this question, we are going to go on a short break of 2-3 minutes where we are going to talk to each one of you here, some of you here. I will leave that to Mahira. So we are going to have a small chat here while we move on to the next section. Everyone has answered this one? Okay, everyone has answered one. Let me just check the chat box. Great, fantastic, great,
Ma'am, I
very
think
well
I'm
done.
gonna have to leave the meeting right now.
Sorry?
Yeah, I have to leave the meeting right now, real quick. I'll just have break and then come.
Sure, absolutely, no problem.
Thank you, ma'am.
Okay. Okay. Now we are going to chat with you, all of you, a little bit. Is that okay, Mahira? Can we do a little bit of chat?
Absolutely. Absolutely. Yep,
Yes, we
I
would
think
want some questions from you as well.
Absolutely, ma'am.
Okay.
Miss Majesta, could we pause the screen share for a second so we could see
Yes,
everyone
absolutely.
more clearly?
Absolutely.
The presentation, yeah.
Yes, yes.
Wow,
And
all right, guys. Oh my God. What an audience, right, Miss Vitasta? I never expected them to be this engaging.
even
Really?
me, it's fantastic to see so many happy faces engaging with the with the session so much and enjoying it actually. It's amazing.
It's amazing, exactly. So guys, so far I think I've heard a lot of you, many of you, but I'm gonna look at a few of you from whom I didn't hear much. Right? I think we have Taha. Hi Taha. I know you're in the meeting and I know you're enjoying it. Would you like to share a bit about what you think? Where did Taha go? Oh, he's here, right? Okay, I think he just left. You're mistaken. No, he's here. Okay, he's gone. All right. Apart from that, I think I'd like to hear from a few of the others who I know. Kasaki, you here? Yasir, I'd say. Hi, Yasir. I know you can hear me. How are you?
Hello, can you hear me?
Hi, how are you? I think you're very relieved after your exams, right?
Yeah, for sure.
Very much, exactly. And I think we have a few new faces, so let me just ask a few questions from them. Can I hear from Vishalakshi? I'm sorry, darling, if I'm not pronouncing your name right, or is it Vishalakshi? I think she's new. She's new here to Utopia, so I would like to hear from her, or even I think I can see someone else as well. Am I speaking to who is this? Miss Saisha, Saisha Gopalan, if I'm not mistaken.
Yes, ma'am. Hello.
Hi, darling. How are you?
I'm doing good. How are you?
Likewise, thank you. How are you finding the session so far?
I'm having great fun, because I'm actually in grade 12, so I've learned
Oh,
about these things, but it's
okay.
actually a lot more fun when ma'am explains it. And I'm actually getting a lot more questions in mind, and I love how she explains it, so it's great fun.
Thank you so much. Thank you.
Thank you.
Well, I'm happy you're having fun. Sorry, Miss Vyasa, you want to say something?
Yeah, I'm saying it's lovely hearing from you. Put down your questions on the chat box. Let's see if you can answer a couple of them, right? Also, everyone, if you have some questions about the session, about us, about Tutopia, please put them down in the chat box. We would be very, very happy to answer those.
Absolutely. And this last person I think I'm going to ask a question from is from Miss Ishita. Can Ishita hear me? Nope. Okay. Then I think, yep. We can go back. Does anybody else have anything that they'd like to share?
Geeta, would you like to talk about something?
Oh,
You have
yeah.
been here for some time. Geeta is from our in-house team. So Geeta, okay.
Me?
Okay.
Hello. Hello.
Yes, Geeta, how are you
Am
finding
I
the session?
audible? Yeah, it's very interesting actually. Lot of the things I didn't know, even I was a bio student myself. But all the intriguing and interesting like tidbits of all these cells and all, it's very interesting. Even like for a grown up like me, I'm not a student, but then also it's quite interesting to hear all these things. And you explained so well actually. I loved it. I am loving the session.
Thank you so
Actually,
much.
actually, I have a
Thanks a lot.
question. Like,
Yes.
when is the next masterclass like this? How-
Okay. So that question, we got to arrange the next masterclass, and you are definitely on the invite list, right? That's something I can tell you. I think Sarath also has the same question. Adan says he has really enjoyed it. Okay, great. Okay, so I think Mahira, should we get back? Should we start again? Or are we? I think we are waiting for, I think, two more participants. Other chapters. Maya, do you want to put it in in this box? Also,
Yes.
I wanted to tell you something really cool now. Now that's break time. We can talk a little bit about that. I think when you spoke about learning for other chapters, right? Also, just give me a second.
Alifia Murtuza, right?
Yeah.
Yes.
Yes.
So,
Alifia. Sorry. Yes, Vitasa. You can go ahead.
yes. So,
Yeah.
like the kind of slides that you are looking at, right? And if you like the fun facts that you're learning here, more interactive learning, do check out our mini learning courses on our learning platform. After this session, you are getting a 1 week free access to the learning platform. You know that, right? Alongside certificates for participating in this. So you are not going out of this in an empty hand. You're getting certificates. You're getting a 1 week free access to the learning platform. And there you can explore many learning courses. It's a very new product of ours, amazing visuals, very interactive. You can learn a lot of fun facts there. There are a lot of mythbusters as well. Mythbusters, fun facts. Did you know there are so many think deep questions? There are so many quizzes to solve, so do explore our learning platform. Okay. And I think Mahira will help you with the access of the learning platform, right, Mahira, post the session?
Yes, I'll be happy to. And I think we also have Yusuf. Yusuf, is there something you'd like to share, darling?
Yes, miss. I am really enjoying this. There are some things that I never knew. I'm just now hearing. It's very interesting to hear.
Oh, thank you, Yusuf. I can hear that it's coming from a very genuine perspective. I really appreciate that. Thank you so much.
Welcome, miss.
Okay, and we have one more person. I'm so sorry, Miss Vijasta, just a second. I'm getting a lot of inbox messages. I'm just trying to give, as many
Absolutely. I'm also loving listening to the stories here, right? I'm also, while I get a bit of break and drink some water. So give me 2 more minutes. It's okay.
Yeah, I think Tarun, is there something you'd like to share, loves?
Yeah, so I'm Tarun. Like I'm actually from Qatar. Now I'm coming. I'm over here due to like holidays, right? So here, yeah, I'm actually enjoying the section a lot. So I already learned this because it's like I already passed 12th. I'm already past 11th. So I like them how ma'am teaches it. So it's kind of more interesting and it's like how to say it's more of knowledgeable section. And actually, my
Absolutely.
passion is also to become a microbiologist. So I'm also on my idea to become a microbiologist.
Oh, wow. Well, Miss Vitasa here holds a master's in microbiology, and I could tell she's loved her journey. So, Tarun, if you
Yeah, it was
are,
like a long goal
watching
passion.
it,
Like I have a side idea to pursue microbiologist if I don't get medical. Yeah.
okay, great. If you're aspiring to be someone like Miss Vitasta's microbiologist, I think part of it, here you go. This is how much you can enjoy the field if you really pursue it.
Yeah.
So, yeah.
Thank you.
Shaik has something to say, right? Yes, Shaik?
Ma'am, science is like one of my most favorite subjects.
My favorite subjects? Are you asking me
And
about my favorite subjects?
no, science is one of my most favorite subjects.
In
And
science?
I'm like the learning things. Few of these things I have heard before, but many of these things I have not heard before. It really amazes me.
Okay. Thank you so much, dear. Thanks a lot. It means the world to me. Thank you so much. I think, Mahra, we are gonna get started once again. Is
We
that
can
okay?
resume. Yes, we can. Yeah. Vitasta, you're on mute, I think. Yeah, we can get started. Right.
Okay. Yeah. Is my screen visible to everyone? Yes. Okay, great. So the question is, if the cell membrane is the only outermost protective layer, is it? Is it, guys?
It is.
Or is there something else? Yes, Fatima?
It is not.
It is not. And which is the other layer?
The cell wall.
Yes, but it's only present in plant cells, right? Okay. Now, if you think about the cell wall, it is compared to as I mentioned, the cell membrane is very stretchy. The cell wall is very, very sturdy, something like this, okay? It cannot bend or it cannot move, whereas the cell membrane is more of like a balloon-like structure. So the cell wall basically gives a lot of strength and very less flexibility, whereas the cell membrane gives less strength and more flexibility. Cool? Yeah. Now, then the question is, why do plant cells need cell wall and why do animal cells not need a cell wall? Or what would have happened if we had a cell wall? Anyone wants to answer that?
Ma'am, if animals also had cell walls, it would like it would be harder to like kill them, and like
Yes.
for other predators to kill them.
Ah, that's such a cute answer, right? It is, and it's true actually. Animals can move away from predators and run because we do not have a cell wall in our cells. If we had cell wall, our cells would have been very sturdy and all very strong, cannot move, immovable like plants. So we would have been stuck to one place like plants if our cells had cell walls. And this is the cell wall is the reason that vegetables are also hard. Have you, and this is something that's I think very relatable for Indians here, but it is the fact that when we buy vegetables from the market, have you ever seen that we try to understand if the vegetable is tender or not? Here, a lot of people go to the market and try it out. I see Mahira smiling, so I'm very sure she has done it. So the tenderness of the vegetable depends on the tenderness of basically the composition of the cell wall. So the cell wall has cellulose and pectin. Okay. So if there is more cellulose and less pectin, the vegetable tends to be really hard. But if there is more pectin and less cellulose, those are the kind of tender vegetables that we would want to buy. Learned a cool fact? Great. Okay. So I have given the answer to this question right now. So can we put the answers in the chat box, please? Yes. Are we getting the answers? I already shown the answer. I think I'm so sorry, guys. I by mistakenly I showed the answer. I'm so sorry. I'm so sorry. Okay, you will have more answers too. Okay, now the next question is about the cytoplasm. What fills the insides of the cell, right? And let me show you how it looks like. Sarat, can you please help me here with the camera? And can you please focus here, Sarat? Yeah. So the cytoplasm is somewhat like this. Can you see? It's jelly-like. And within the cytoplasm, you can think of these colored balls as the cell organelles. Now, why are these so small? Because compared to the cell, the organelles are very, 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 very tiny. Even though the cell is very tiny, it is microscopic, cannot be seen with the naked eye. But the cell is something that you can see with a compound microscope. But the organelles are something you cannot see without an electron microscope. That is a much higher magnification. Okay? So the cytoplasm is something like this, jelly-like. Can you see this? This is the consistency of the cytoplasm. Okay? And within the cytoplasm, we have the nucleus and the different cell organelles. Okay? Great.
Miss Vichasta, can we maybe bring it forward a little bit so that I think everyone can see it if the screen is...
Yes. Can you bring in the camera a bit? Yeah. Here, can you see it now? Is this better?
Yeah. Yes.
This is the consistency of the cytoplasm. Okay, great. Can we have a tissue, please? So that I can wipe my hand. Thank you so much, Sarat. Thanks a lot. Okay. Now, these are the different functions of the cytoplasm. It gives shape to the cells, it holds and protects the cell organelles, and it stores the molecules required for the different cell functions. Both animal and plant cells have the cytoplasm. Okay. Now tell me something. You just looked at the cytoplasm. What would happen if you didn't drink water at all? Would anything happen to your cytoplasm? Anyone wants to answer that?
Ma'am,
Yes.
our cytoplasms would get destroyed by themselves because our cells needs water to function.
Yes. Exactly. Absolutely correct. If there is no water, if we do not drink enough water, all our cells will get dehydrated because the cytoplasm is 70% water. And that is why our bodies are 70% water. Right? Great. Now, these are the different cell organelles. Okay. Now, we are going to move this side and bring this here so that we can show the different cell organelles. Can we please move this around? Yes. And can we place it here? Can you yes. Yeah, we can get this started. Okay, so this is our cell. This is the DIY cell actually. Okay, and we have the different cell organelles here. Now, if we talk about this is the endoplasmic reticulum. There are two types of endoplasmic reticulum. Can you bring the camera a little closer? Yes.
Yeah, Miss
So
Vijasa,
one,
if you don't mind, could we stop the share screen so we can see the model much better? Yeah.
yes, sure, Mahira. Just give me a second here. I'm so sorry.
Guys, this is the ultimate, I think, such an amazing, beautiful 3D model. You can envision the cell completely using this. So here we go.
Yes. So the ones that you see are with the ribosomes, the thermocol balls, these are the rough endoplasmic reticulum, and they help in protein synthesis. Now, why are they rough? Because they're studded with ribosomes. The ones here are smooth because they do not have ribosomes, and they help in lipid synthesis. This is the nucleus. This, the one that you see here, the red one, this is the mitochondria. It is also called as the powerhouse of the cell. Why? Because it helps in the generation of ATP. Now, this is the mitochondria, which is like not cut open. And here we have a cut open mitochondria, where you can see the insides of the mitochondria. Yes, Shai, tell me.
And are there two powerhouses in our cell?
We have the mitochondria is the powerhouse of the cell. Yeah, so the depending on the kind of cell, for example, muscle cells have a lot of mitochondria because they need a lot of power to help us run and walk, right? So which is why based on the kind of cell, there will be a little more endoplasmic reticulum and a little less maybe mitochondria, a little more mitochondria, less Golgi body. The number of cell organelles a particular cell has depends on its function again. Okay, is that clear?
Okay, clear, ma'am.
Yes, thank you. And then we have the ribosomes. This is the nucleus with the nucleolus inside. So this is an open nucleus actually. Then here you have the Golgi apparatus. So whatever protein is getting synthesized from the endoplasmic reticulum is packaged and transported by the Golgi using vesicles, which you can see here, the yellow colored ones, which pinch up from the Golgi. And then you have the green colored ones, which are your small animal cell vacuoles. Now in plant cells, if we have seen plant cells, in plant cells, the vacuoles are very big and they cover most of the cell. Okay, and apart from this, because it's an animal cell, we do not have a chloroplast here, but with all of these organelles, plant cells will also have plastids, which are of three types. There are I think I'll start sharing my screen once again. Yeah. So we are with that. So I have already mentioned the functions of each of these. So I'm not going to, I'm so sorry. Screen sharing has been stopped. Yeah. So I'm not going to repeat and bore you with the same slides. But let's solve a question here, okay? And then let's move on. Just a second. Yes. Can we answer the question? Do we have the answers? Okay, fantastic. And all of you have answered smooth endoplasmic reticulum A? Yes, fantastic. Now, I've also spoken about the Golgi apparatus, which helps us in packaging and modifying the lipids and the proteins produced by the endoplasmic reticulum. Then we have the lysosome. The lysosomes are actually formed by pinching off from the Golgi. So these are initially small vesicles. And when you go to the higher classes, you will further understand that lysosomes are 4 kinds. But primarily, they contain enzymes which digest foreign material as well as old cells. Okay? So they are also sometimes called as the suicide bags of our cell. Then we have the mitochondria, the powerhouse of our cell that produces energy. Now, one very interesting fact about the mitochondria is that it's a modified prokaryotic cell. Do you know why and how? We are going to come back to this at the end of the session. I would want to learn from you why and how we found out that the mitochondria is a modified prokaryotic cell. Okay, great. Now we have another question. Can we answer this? Find out the correct sentence. Are we having the answers in the chat box? Okay, let me help you a bit. So the rough endoplasmic reticulum produces protein. The smooth endoplasmic reticulum produces lipids. And then these are packaged and modified by the Golgi, then put into vesicles. And these vesicles then later turn into lysosomes. Now, can we have answer to the question? Okay, great. We have some answers. Did we have A as answer? Good, great. Lysosome arises from? Are we having answers? It is B. Yes, it is from the Golgi apparatus. Very good, very good. Congratulations to everyone who answered B. Now, as I spoke about the vacuole, there are small vacuoles in the animal cells I just showed you, but plant cells have larger vacuoles that store a lot of the plant's excretory materials. Then in plants, we have chloroplastids, there are chloroplasts, which helps in photosynthesis and synthesis of food. Then there are 2 more different kinds of plastid. One is a chromoplast and the other one is a leucoplast, okay? So leucoplast, chromoplast, and chloroplast. Leucoplast helps in the production of starch and the storage of starch. Your chloroplast basically helps in photosynthesis, and chromoplast produces and stores colored pigments, which is responsible for the colors of petals and in flowers that you see. Okay, great. With that, we are kind of towards the end of the session. I do not want to bore you with the repetitive slides because I've already spoken about those. So let's move on to the nucleus. Okay, let's move on to the nucleus, which is the brain of the cell or the control center of the cell. And here I have a model of the universe, of the nucleus for you. I'm so sorry. Of the nucleus for you. This is a cut open model. So you can think of the nucleus looks somewhat like a ball, okay, which is placed at the center of the cell, as you can see here, somewhat like this. And when you cut open the nucleus, this is what you see. There are ribosomes. There is this jelly-like matrix within the nucleus as well. Okay, there is this jelly-like matrix within the nucleus as well, which we call as the nucleoplasm. Within the nucleoplasm, what is suspended are the chromatin threads. And this big ball that you see here is the nucleolus, the red thing that you can see here as well. So that is the nucleus, the control center of our cell. It controls and coordinates all activities of the cell and helps in cell division, and which is why it stores all the genetic information. So the reason we look like our parents is hidden within the nucleus. The DNA is hidden within the nucleus. Okay, great. So I think it's time for one last question. Okay. And then I'm going to hand it over to Ms. Mahira and she can take it from there. Okay, we need to write down the answers for these questions here. And after you answer this question, Miss Myra is going to talk a bit, and then I am going to talk about a bit about the different careers that you can have in cell biology if you're interested. Okay, are we having the answers?
Ma'am, excuse me, I have a
Yes.
question.
Yes, tell me.
Is it supposed to be which or is it supposed to be where?
Which kind of plaster is more common in the roots of the plant?
Oh,
So
okay,
you
I
have
get it.
to
I get it
mention the
now.
name of the plaster. That's yes, right.
Okay,
Absolutely.
yeah, got it. Thank you.
So I think we are going to stop this, like the video sharing from this one. And yeah, have we gotten the answer? Did we get any answers? We have roots of the plant, leucoplast, leaves of plant, chloroplast, plant. Great, fantastic. Sharanya has answered, Elizabeth, leucoplast, chloroplast, fantastic. Okay, B is chloroplast. Yes, so the correct answer A is roots of the plant is leucoplast, leaves of the plant is chloroplast, and fruits and flowers is chromoplast. And with that, this session, this masterclass, kind of the content of the masterclass kind of comes to an end. But we all obviously have certain things to talk about. I'm going to pass it on to Miss Mahira now, and then I'm going to take back again for some career guidance for all of you.
Right. Thank you so much, Ms. Vitasta. I think that was a lot, guys. How are you feeling now? I think it was like, a lot of things. I think what I feel is, I personally start with myself. I think biology is so much more than what we think it is, because a lot of us, when we study biology at school, we tend to think like, oh my God, too much to by heart. Oh Lord, a lot to keep in mind, like so many new terms you can't even pronounce. Like, how do I get through these new words? Especially human biology and cells is like a totally new dictionary, sometimes it's like Greek. But then the moment you realize the fantasy, the kind of theory behind it and how much interesting it can be made, then you develop that passion, don't you think, guys? Is there anyone who would like to share their opinions on that? I think some of you guys I already know. I know what you think about biology. I'd like to invite a few others who would like to share their views. What do you think? Guys? Alishwa. Hi, Alishwa. I'm seeing you after quite some time. How are you? Alishwa? Okay, I don't think she can unmute at the moment. Can she? Ali? Hi, Ali. How are you?
Yeah, I'm doing good. How are you?
Alhamdulillah, I'm doing great as well. Thank you, Ali. Thank you so much for joining. Ali, would you like to tell me about what you think and how much, I don't know, if you really felt the whole passion about biology from this whole masterclass?
To be honest, I don't like biology a lot, but from what the teacher taught us today, I was kind of interested in all of the information. Like I've heard a lot of new stuff that I haven't learned before from school or studying alone. So it was a nice session.
Thank you
Thank
so
you,
much.
Ali. Thank you so much.
Thanks.
Guys, Ali has been one of our really good students from group classes, I must say. Not only him, there are so many others, especially, I mean, if I go to mention names, it's going to be a whole long list. But thank you, Ali. How about Reem? Hi, Reem. How are you?
Goodness.
How have you been, Reem? And how did you find the masterclass today?
It was very interesting.
Okay.
So I think we are going to share a Google form with you right away
Right
on the
now.
chat box. And if you want the new masterclass, another masterclass. And if you really enjoyed it, please do fill up the Google form and let us know. It is just very few questions, very short. So we are going to do, as you fill up the Google form, I'm quickly going to give you some tips and tricks on your career front if you love cell biology, like I do, clearly. And which led me to take up microbiology. And I think someone mentioned here that they would want to be a microbiologist. And it's amazing when you study microbiology. It is just, there are around 21 subjects that you study alongside, and all of them are equally interesting. So you study everything from biotech to genetics to immunology to biostatistics to biophysics to biochemistry to genetics to molecular bio to microbial genetics. There's so much to learn from, and it opens up a whole new arena, and then you can specialize in any one of these. Obviously, if you love cell biology, there is the traditional biotech and microbio, but I want to talk about something that's a little different. That is the field of pharmacology, right? You can be creating drugs. You can be the one to design medicines and cure cancers, right? You can also be a cancer biologist because, or not an oncologist. An oncologist is a doctor. So if you pursue your sciences, you're studying the sciences, you do your grad and your post-grad, you can also be a cancer biologist. You can be a genetic counselor. You can help people understand their genes and their DNA better. And by being a genetic counselor, you can also develop personalized medicines. That means that a particular medicine only designed for a particular person, right? So there can be personalized medicines as well. So that is something that you can do. And apart from that, definitely you can teach. You can work like me at EdTechs for sure. And obviously there you can do genetic engineering. You can get into the field of biophysics or biostatistics, where there is a lot of data computation involved. You can actually do bioinformatics as well, whereby you study the structure of proteins and correlate it to the outwardly the physical features of organisms. So there's so much that you can do with cell biology if you love it, honestly. So yeah, and now it is almost, we are kind of about to end the session. So before we do that, Mahira, a huge shout out to you. I hope you are here in the call, Mahira. I am unable to see you, but I hope you are on the call because... Okay, so a huge shout out to Mahira for helping us do this, for arranging this, for hosting this, for getting all of you, inviting all of you, for sending out reminders, for being my constant boost and my constant companion throughout this journey of crafting the masterclass. It was a wonderful, wonderful experience working alongside her in this project. Alongside that, I will have to thank Sarat. Today, Sarat, I would want you to come here and say hi to everyone. He was behind the scenes all this while, working with the camera, with the props. Hi, guys. Say hi to Sarat. So Sarat is... Yes. So
Hi.
that's Sarat. And then we had... Yes, Shai. And then I have to thank two people outside the organization as well. So Mahira, were you there all this while I was speaking?
I'm so sorry. Yes, I think I
Okay,
just
I'm
had
going
to
to
get,
repeat that for
I was
you,
actually,
for sure. I
kicked
was
out of
doing a bit of credit section here, right? And I was telling everyone
Oh no.
how lucky I am to having the opportunity to work alongside you. The amount of your contribution to this masterclass was immense. You just worked day and night
Thank you.
with a lot of passion, helping us craft this, nail this. If it was good, 60 to 70% of it is because of her. She invited all of you to join this if you all enjoyed it. So 60 to 70% of the credit goes to her for sure. She is amazing.
Oh my God, that's an amazing amount, but yeah, thank you so much. I'm humbled.
And I also introduced them to Sarat, who was behind the camera, helping me with the props, helping me with everything here, handling the camera. So big shout out to Sarat once again. And there are a couple of friends and family members from my end as well who helped me with this. So you know who you are. So I'm not going to, because they are not in Tutopia, I'm not going to take names. So, but thank you so much for that. You know who you are. Hopefully you're there in the call as well. So yeah, that's from my end, Mahira. Over to you. Thanks a lot. I had a lovely time
I
interacting with all
know.
of you.
Guys,
And
I'm gonna spill a few beans here, right? It wasn't like it wasn't very confidently at the start. We were so on edge, on our toes, like super nervous. But then thank you for being such a wonderful audience, I must say. Thank you for all your efforts in really making this more interactive, more engaging. And out of all of us, if there is anyone who really, really deserves hats off, a standing ovation would be Miss Vitata Datta. I think she's gone beyond, out and beyond her way, guys. So if you would really want us to continue this, we need your support, right? So therefore, I've sent across the Google form. Please do fill it out. And yes, as you know, our gratitude, show our gratitude for joining the session. We will be given 1 week's free access to the platform. Ms. Vitasa, I'm going to take a quick 2 to 3 minutes to take them through the platform. Is that all right?
absolutely. Please feel free.
Yeah. I'm just going to take a quick 2 to 3 minutes, guys, because you all have to first know how to use a platform, right? So firstly, I think, thank you so much, Geeta. Firstly, I think once you get access to the learning platform, you're going to have a student account at Utopia, which is one of, all you're going to get all the first-hand experience of how user-friendly our interface is going to be. So you're going to be given student accounts at Utopia. So when you get the account membership, you're also going to be given free access to the learning platform, right, for 1 entire week. So how are you going to go about this? I'm just going to give you a quick tour so that when you all get your access, you all know where to go, right? So let me share my screen and we can get started. All righty. So can you see my screen here, guys? This is my account, right? So here we go. This is how you're going to see it if you're a student. So you're going to click on the learning platform and you're going to get directed into our treasure trove, right? So here you go. So why a learning platform, right? What is the purpose of it? If you're someone who is looking for easily accessible, anytime, anywhere resources of high quality standard and engaging content, here you go. So the learning platform has resources across several curricula and catering to several different subjects as well, as you can see. So let's take a quick look as to why the learning platform is so cool and amazing, right? So here you go. If you're an IGCSE student and if you are particularly interested in the subject of mathematics, I'd say we here have a whole list. Onto my left, you have the list of main topics. So the main chapters of the syllabus have been listed down, and then onto my right, you have the breakdown of its subtopics. Yeah. So let's take a deep dive as to what we have in the learning platform. So we do have 3 main parts of it. I would say resources. So what we mean by resources are detailed theory notes, as you can see, right? It covers the entirety of this chapter of the syllabus. So if you are a person who learns on your own, this is a one-stop solution for you. So you can read through our theory notes, our resources. And if you are someone who is benefiting more through a video explanation, we also got you covered. We have videos under every chapter that you want to study for. So you click on the video here and it will direct you to an understandable video explanation of the difficult theory concepts, right? And let's say now you're done with this topic and you want to test your knowledge. You want to see how competent you are with that particular topic. So you click on attempt a quiz and you're directed to an engaging quiz arena here. So when you click on start a quiz, you will be having MCQs So what you're going to do is select the most apt answer, the correct answer to each of these questions. I'm just going to take you through a quick tour. Therefore, I might skip a few questions. Please excuse me. So here we go. We click on the right answer that we think is the best and submit it. You also get a pop-up here just to see, you have 6 unanswered questions, Mahira. Are you sure you want to submit it? Yes, I am. So we click on submit and voila. So this particular question, yes, I've got it right somehow, thankfully. And we've got AI integrated into the platform that will tell you, Mahira, your answer is right. And this is your stepwise solution to the answer, along with the marking scheme. So what is the personalized feedback you can have? This is your personalized feedback. And what are the suggestions for improvement? So it goes on this way. And let's say, sadly, you didn't get your answer right. We've also got you covered because AI can tell you why your answer is wrong. And it will also give you the right steps to follow for the correct answer. So this is how we've structured our learning platform, guys. And if you want to come back, you just navigate very seamlessly through the platform and it will bring you back to the main page. So not only do we have resources, videos, and quizzes, we also got assessments. So since we're discussing biology today, let me take you through how we have assessments in biology, right? So like Ms. Vitasta correctly said, we have something exclusive called the mini learning courses. So you click on assessments here and you'll be able to see all the different types of assessments that have been mentioned down. And what's so interesting about these assessments, these are mock exams that are generated through AI, right? So the quizzes here, the resources here are all the kind of questions that have been analyzed through AI for the past 6 years, so that there is a good flow to the questions. The question patterns have been analyzed. Here, I'm just going to give you an overview as to how we generated the AI mock exams. And it will also give you a good coverage, right? Some of the questions you may not have encountered before, some of the questions may be new, but it's a learning curve there. It's a learning process. So you can learn through a vast I think, coverage of differently styled questions so that you all can gain more insight on what are the different types of formats of questions you can expect. Not only that, we provide the marking schemes as well. I don't think I have the time to go deep into them. I'm just taking you through it, right? So here we go. These are the assessments you can take. And if you want to know where exactly you stand in terms of your academics, we have something called the diagnostic challenge, right? So the diagnostic challenge is a part where you'll be able to figure out, right, so if I am good at this particular subject, then can I get a detailed analysis? Can I get a report of, what are my strengths? What are my weaknesses? And can I also get a place, a solution of the kind of academic plan I should be following? Right? If I am in this particular standing of the subject, it's something you can gain from our diagnostic assessments. So I've attended the assessment. I've submitted the answer. I think my internet connection is giving a bit of trouble. So I'm going to wait until the answer is submitted. So here I'm getting my score since I've just randomly guessed. Please don't judge me. I'm here getting only 1 out of 10, right? So something that we really, really want to promote to Tutopia is once we have attempted the diagnostic assessment, it can give you a good overview of what your strengths are, what your weaknesses are as well. I'm just going to wait a little bit. I think due to my connection, it's taking some time to load. But yes, if this is something that you will find interesting, this is something that you will get to experience firsthand through Tutopia's learning platform, for which you'll get free access for an entire week starting from tomorrow onwards. So if you want to really get started by tomorrow itself, I will be sharing the login details with you guys. I'm sorry, something seems to be wrong here. So anyways, let me come back to the home screen. And if you want to come to the section where you want to test out how well you performed in your quizzes, we have the quiz history as well. So the quiz history will give you a breakdown of how your progress, right? So what are the kind of, for example, if you've made attempts, then how much have you scored in each of the attempts? What times have you taken to finish it? All that is mentioned here, right? Okay, so that's my tour for you guys of the of the learning platform. This is once again your student account. Under the student account, there are several tabs here. You can just play around, figure out how to use the platform, and we can see Masterclass. I'm hoping we can have it very soon. So if you finished with your feedback form, then I think we can figure out when we can have it next. All right, I'll pass it on to Miss Vitas that she has very few words to say at the end, and then we can wrap up.
I think this was a wonderful session. I mean, I had so much fun. I actually interacting with all of you. I have been, I'll be very honest, I am taking this like a class after a very long time because I usually work in edtechs and been in the background primarily. So it's been a very fruitful experience and a very nice evening for me, interacting with all of you, learning together, exploring together, learning more about cells, and hope this session inspires you and inspires the love for cell biology in you. And you're also going to get your certificates by next week. By Monday, Tuesday, you're going to get your certificates. So, and if you want more such masterclasses, do fill in the Google form. We would really appreciate that because it's only your support with which we can have more masterclasses like this. Thanks a lot. Thank you so much. And Mahira, thank you once again for being such a wonderful host.
Yeah, Miss Pitasa, we have a request about the slides that were shared today. If we can have it shared.
Yes, absolutely. We will.
I think there was so much effort put into making the slides. So yes, I think we'd be more than happy to,
we will share
help
it.
you learn.
We'll share the slides alongside the recording. I think Mahira, some of them joined late. Yes. We can share the recording.
The recording as well. All right, guys. I think I'm gonna be like a typical host and be like, see you soon, and thank you for staying tuned throughout the whole. My word, 2 hours. It has been 2 hours, guys.
Yes.
Thank you so much for being such a brilliant audience.
Thank you so much.
See you soon in the next masterclass.
See you. Bye. Thank you, everyone.
Thank you, Miss. Have a good day.
Have a good day. You too. Bye.
Bye.
Bye, Adan. Bye.